So good morning, good afternoon, and good evening to everyone. And welcome to this, our first joint webinar with the Plant Biotechnology Journal. Before introducing our chair for today, Professor Johnathan Napier, let me share some basics on the logistics of this meeting. The session today will last about 90 minutes and will include two presentations by Drs. Edgar Cahoon and Hyojin Kim. Feel free to use the chat feature to speak with the rest of attendees or with us, the host and speakers. But for the Q&A session, we will use the Q&A feature in the Zoom toolbar that you may already know. There you can send your questions and about your favorites, so that you will help us during the Q&A session that way. This Q&A session will happen at the end of the session once both speakers have finished presenting. So let me introduce our chair for today. Johnathan Napier is the editor-in-chief of Plant Biotechnology Journal and a professor at the Rothamsted Research in the UK. He is a leading figure in the plant biotechnology and advocate for the public good potential of GM crops and an active voice in science communication, such as right now. So Johnathan, the floor is yours.
Okay, thank you very much, Isabel. I really appreciate the efforts that you and the Global Plant Council have put into organizing this joint webinar. I think it's a really good initiative, and on behalf of Plant Biotechnology Journal, I'm really glad to be involved in this, which is the first, I hope, of many such webinars that we'll do in 2026. So yeah, I should just remains for me now to introduce our first speaker, who is I should declare an interest and say a great colleague and collaborator and a friend as well. I'm really pleased that Ed and Hyojin, who you'll hear in a minute after Ed's spoken, agreed to give this seminar. But Ed is, I'm going to, if it looks like I'm reading from a piece of paper, I am reading from a piece of paper. I just want to make sure that we get all of Ed's accolades onto the record. So Ed Cahoon is the director of the Center for Plant Science Innovation and the George Holmes University Professor of Biochemistry at the University of Nebraska, Lincoln. Ed conducts basic and applied research, which is really a key message, I think, from the webinar that we're going to hear today. So Ed conducts basic and applied research on plant lipid metabolism, and the goals of his research are to enhance the nutritional and industrial value of crop plants and to probe the synthesis and function of bioactive lipids for nutritional biofortification and improved agronomic performance of crops. So with that comprehensive introduction, we will turn over to you, Ed, to give your presentation, which I'm very much looking forward to.
Okay, thank you, Johnathan, for the introduction, and thank you, Isabel, for having us today, Hyojin and I. And so let me get this all set. Oops, it's already my second slide. Okay, 1 second. And I'll remove the... Okay, so thank you all for being here today and to have this opportunity to tell you about the research that we do in my lab and also in the Center for Plant Science Innovation at the University of Nebraska, where we take fundamental knowledge and try to translate that into real-world impact. And I'll give you kind of an overview of what we do, and then Hyojin will give you a very specific example that was recently published in the Plant Biotechnology Journal in the August edition. Okay, 1 second. Okay, so as plant scientists, I think we have the opportunity to really address major agricultural grand challenges. And two of the major challenges really revolve around food security. Of course, we want to produce more crop per hectare. And we also want to think about how we can protect yields. That means how can we make plants more resilient against drought, heat, and so forth, and also more resistant to pathogens like insects, bacteria, fungi. And so these really are related to food security. We also recognize that that if farmers don't make profit, if they don't make money, we won't have an agricultural sector. And so we also think about a grand challenge as being increasing crop value. And this might involve making more value or new markets for farmers to make additional income beyond these large commodity streams that we have for our major grain crops. And so the enhancing yield, protecting yield, these really fall under the category of input traits. They're related to production. Output traits are the increasing the plant quality and marketability. And so the major seed companies like Corteva, Bayer, Syngenta really focus on the input traits. This is where they see the profit, producing more per acre or maintaining the yields. But really, we see that there is an opportunity on the output trait side. And this may be overlooked again by the large companies and really provide an opportunity for innovators, for entrepreneurs. And if there are young people in the audience, maybe this is an area that maybe you should be thinking about. And you could maybe think about how you can take your product and start a company, or how you can license your new technology. Okay, and so when we think about innovation, the most important thing is gaining knowledge. And as I say here, knowledge is supreme. And so we have to, before we can have impact, we have to know things and the basic functionings of plants. And so typically knowledge is derived from model systems. Either the knowledge is discovered or validated in the model systems, might be Arabidopsis, Benthamiana, or microbial systems. And so when we think about this, okay, this is typically, we do this, make this discovery, we make the big discovery, we publish the paper in a good journal, and we're very happy. And that's typically where the project ends. Okay, so really for knowledge to have impact, it really needs to be translated and used for something. And so that's what we think about. Okay, and I would say that many of the discoveries we make in model systems, what we find is that they don't always translate into the crop and what we see in the field and when we apply it. Okay, so many of us will then translate this knowledge, this discovery, into a crop, and then we evaluate it in the greenhouse, and we call it the end. That's the end. We publish our paper in Plant Biotechnology Journal, and we're all happy. But really, if we want to get to a product, we have to think about the whole pipeline. And then labs like mine and Johnathan Napier's in the UK, we think about how we can go from the greenhouse to the field. How can we evaluate the traits in the field, ideally over multiple seasons and in different locations? And with this, what we find is that what we see in the greenhouse, it's really a pampered environment for plants. The field is the real world. And often we see that maybe the trait, it doesn't really hold up in the field, or maybe it's better than we think in some cases, actually, when we take it to the field. And in this talk today, I'm going to mention how we can go from the field to application, which it really involves functionality evaluation. How do we gain information? And we're going to focus on products rather than input traits. And how can we do the evaluation to see that what we've done, what we've made, really has some value? And you can see that these arrows are getting narrower and narrower because fewer and fewer people do them. And at the end, there are very few products that come out from the translation of knowledge. And we'll talk more about that at the end, why that is. And you can see some examples of like the Plenish high oleic soybean from Corteva, the Norfolk produce, the purple tomatoes, Aquaterra that's producing fish oils and canola. These are just some examples. Okay, and so yeah, what I want to emphasize here is that not only do we gain information about the product we're trying to make by this pipeline, but also there's feedback that goes into this. And so we learn from each of these steps. And often what we find is that maybe the knowledge can be refined, maybe we need to make another discovery. We need to generate more knowledge because what we're trying to do actually doesn't work that well. And Hyojin, I think in her talk, she'll tell you more about that. And this really gets into this feedback loop and thinking about in synthetic biology, this idea of design, build, test, learn. And we'll hear about that, 2 iterations of that in Hyojin's talk. But that's the idea that we design something, we make it, we test it in the field, we evaluate it, and we learn from that. And that's one way to gain additional knowledge that can really feed into this pipeline and improve whatever our product design is. Okay, so if we want to translate this knowledge to crops, what we are actually doing is introducing genetic variation. And so genetic variation can be generated through multiple means, breeding, gene editing, transgenesis, putting trans genes into crops. For this talk, we're going to focus largely on transgenesis. And the great thing we think about transgenesis is that if we're trying to make a new trait, some high value trait that's not ever been seen in a plant before, we can tap a large amount of genetic variation across plant species, across organisms to affect the change that we want. But we should also keep in mind that transgenesis, breeding, and gene editing, these don't function in isolation, that even if we have the best product, we want it to be in the highest yielding background, the most resilient background, for example. And obviously, it's not just about the introducing genetic variation. There's technological advances that are really fueling this pipeline that are really making the translation much faster and more precise. Hopefully we'll get to a point where there's tissue culture-free editing and transgenesis. Progress is being made on viral vectors, for example, synthetic biology, AI, machine learning, high throughput phenotyping. These are all kind of feeding into this translation pipeline, and I think the future is bright. Okay, so I want to shift gears a little bit and tell you about the work that we do. So you probably, maybe some of you don't even know what Nebraska is. It's in the United States, and it's actually in the middle of the United States. So if you point your finger, close your eyes, point your finger on the map of the United States, it's right in the middle. And it's actually in terms of agricultural receipts, it's the number 3 state for agricultural receipts. It's number 1 part of our economy. California is number 1, Iowa's number 2, we're number 3. And so in production of crops like soybean and corn, we're among the nation's leaders. And our state actually is, we call it a living laboratory for water relations, because in the eastern part of the state, there's a lot of rainfall, like the Midwest. As we go to the western part of the state, it's dry land agriculture, and we can test crops over both kind of climatic zones. And so we think that Nebraska really provides a real world proving ground for the type of research that we do in the Center for Plant Science Innovation and in my lab. Okay, so at the University of Nebraska, I direct the Center for Plant Science Innovation that includes 22 faculty members across 5 departments and 2 colleges. And we have a large focus on fundamental knowledge acquisition, gaining fundamental knowledge, really understanding how genetics, environment, evolution affect crop yield and quality. But we also think about the grand challenges that are faced in agriculture and society. And we think about how can we take our discoveries and translate them, and then maybe at the end, maybe make a product that really impacts food security, profitability, and so forth. And so key to this is the ability to translate from discovery to crops. We do this by biotechnology and breeding. We have field facilities, high throughput phenomics facilities, and so forth. And so you may not have heard of us, but our biggest product actually is one of the major biotech products in the last 15 or 20 years, and that's the dicamba herbicide resistance trait that was discovered by Don Weeks and Tom Clemente in our center. And this is now in 50% of the total US soybeans. And our university licensed this to Monsanto and now Bayer Crop Science, and this brings back money to the university and to our center. Okay. So we also think about how we do our work in terms of an integrated biotech pipeline. And this is just showing an example with soybean, where we develop vector systems, and we have in-house capacity to do soybean transformation and transformation of other crops. We do the greenhouse evaluation. Then we have dedicated field sites for biotech crops where we can evaluate if it's an input trait, how well the trait holds up, how well the crops perform in the field. If it's an output trait, we can harvest maybe seed in the case of soybean, and then work with our industrial ag product center to extract either the protein, the oil, the carbohydrate, and evaluate that in different tests. And so you can see that the types of traits in our pipeline are both our input and output traits. And I have to say, I think that we were kind of at the University of Nebraska somewhat at the leading edge of crop biotechnology. So our former director of the Plant Transformation Core Research Facility, Tom Clemente, started transformation capacity at our university with crops very early in the 1990s. In fact, he recently retired, and we've transitioned to a new director, Nathan Butler, who oversees this biotechnology pipeline. We, of course, do model plants, but we also do the major crop plants we also transform the major crop plants of Nebraska, maize, wheat, sorghum, and soybean, and also rice, which is not a major crop. But Nathan also does fee for service, so please reach out to Nathan. That's his email address if you are inspired to translate your knowledge from Arabidopsis to a crop. And so in addition to the transformation facility, as I mentioned, we have 2 dedicated sites in the state for evaluating biotech traits. One's in the eastern part of the state, one's in the dry land western part of the state. We primarily in my lab use the location in the eastern part of the state. This is just examples of camelina and soybean and sorghum that have been evaluated. And at the end, in the case of grains the work that we do, we can generate large amounts of grain that we can then use for subsequent functionality testing. And these are all done under USDA APHIS permit. These can be very stringent requirements, especially for sorghum. We could talk more about that at the end. Okay. And for us, we think about what our synthetic biology, metabolic engineering platforms are. And so, many of you are aware of the great work Johnathan has done in camelina. So we also use camelina as one of our platforms. And the reason we use camelina is that it's really easy to transform. It's like if you're a Arabidopsis person, you can do transformation of camelina with floral infiltration. It's a close relative of Arabidopsis, so discoveries in Arabidopsis can be translated quite easily into camelina. It's got a short life cycle of maybe 3 to 4 months. You can get maybe 3 cycles of camelina done in 1 year. And we think about, that maybe it's a good rotation crop with other like soybean or wheat, or even now people are thinking about it as a cover crop. And for sustainable fuels, it has a low intensity score, carbon intensity score, which is a good thing. It helps make camelina more competitive with other oilseed crops. We also work on soybean and sorghum. I just will mention soybean real briefly, that both soybean and sorghum require tissue culture and a lot of labor for the transformation. And so we have to work with our transformation facility to engineer soybean and sorghum. But the good thing about soybean is that it's an established crop in our state and in the United States. And we use it as a platform for looking at aquaculture traits and livestock feed traits. And a really great thing about soybean is that it's a legume. And so it fixes nitrogen. And so there's less impact on the environment, with using nitrogen fertilizer when you grow soybeans. Okay. And I'll give another shout out to camelina. And we say it's a dream crop for metabolic engineers. And you'll hear Hyojin talk about largely her work has been done in camelina, but then also extended to soybean. But as I mentioned, there's no tissue culture that's involved in camelina transformation, and it's not labor intensive, and it really bridges translation from Arabidopsis to crops. For us, it's very predictive of if we do seed traits in camelina, they translate generally quite well to soybean. And I think a lot of us are thinking that now there's availability of winter varieties, and we think having both of these may be game changing. So if you have a product, maybe you can produce it 2 times a year, both in the spring and winter varieties, or maybe grow soybeans in the spring and then come back with winter camelina that has your trait of interest. And if you're interested more in camelina transformation, we have a YouTube video that you're welcome to look at. And in my lab, we largely work on seed traits. What you'll hear from Hyojin is done with seed-specific promoters. We do modular gene assembly. We use golden braid generally for our stacking our genes. And we have selection markers like DS Red for glowing seeds and herbicide resistance with BASTA generally. And so in this pipeline, you infiltrate the camelina. If you use DS Red, you just let the seeds set in the greenhouse, and then you can look for glowing seeds, and those will be transformed seeds. So it's quite easy. And before I end, I just want to give you a few examples of this theme of today's talk, the lab to field application research. And I think what you'll see and what Hyojin and I talk about is that to go to application, you really have to build collaborations across disciplines. So we're working with, for example, chemical engineers to generate sustainable aviation fuel. Hyojin will talk about work that she's doing with food scientists. So that's the great thing about this research, building collaborations, always a great thing. And so this was a paper that was published in Plant Biotechnology Journal earlier in this year in print. And this is work that we've done with sorghum, where we've tried to engineer it to make vegetative oils in the leaves and stems. That's really for renewable fuels and bioproducts. And this is work that's been done as part of a DOE plant a bioenergy research center called CABI, which is the Center for Advanced Bioenergy and Bioproducts Innovation. But the great thing about CABI is that we have a lot of collaborators. And so we developed this oil sorghum that produces oil in its leaves and stems, vegetable oils, basically. And so we then, working with Illinois, we have this feedstock to fuel pipeline where we grow the sorghum both in Illinois and Nebraska over 2 seasons. We then use the material to work with the bioprocessing lab at the University of Illinois that develops methods for extracting the oil from the vegetative parts of the plant. Then further on in the conversion theme of CABI, there are scientists that are working with developing catalysts to convert that oil into sustainable aviation fuel and renewable diesel. And we also think about the DBTL cycle. So we have a level of oil we think we could do better. So we use systems biology, transcriptomics, fluxomics to really learn from this oil sorghum, this generation, and try to develop a second generation through the DBTL cycle that produces more oil. And this is just showing here work that we do with a chemical engineer at Northeastern University who's developing new technology for making jet fuel, basically making alkanes from vegetable oils. And his method really requires a mixture of short and long chain fatty acids to get a fuel that looks like the alkanes that you find in sustainable aviation fuel. This is kind of our DBTL cycle where we transform the camelina, take it to the field, evaluate it, extract the oil, hand it off to the chemical engineer. He tells us, well, the formulation could be better. And so we try to introduce new modifications that really get to a point where the fatty acid profile of the oil matches his technology. And a final example that is work that we're doing that's part of the grant funded by the Department of Energy, Bigger, Better, Brassicaceae Biofuels and Bioproducts. And in this project, we're really focusing heavily on the knowledge acquisition, where we're trying to understand how fatty acid synthesis is regulated for the chain lengths, for the total production of fatty acids, developing new synthetic biology tools that allow us to better translate the knowledge. And AIM 3 is basically using that knowledge from AIMS 1 and 2 to really generate a product. And so we're thinking about how can we get camelina and pennycress to have oils that are more similar to Kufia that has short and medium chain fatty acids that are really good for sustainable aviation fuel. Okay, so today we're going to hear from Hyojin Kim, and she recently published a paper in Plant Biotechnology Journal that was the cover of the August edition of Plant Biotechnology Journal. And this is work that was done in collaboration with our host today, Johnathan Napier, and his lab, Richard Haslam at the Rothamsted Research. And what Hyojin is going to tell you about is trying to address the need for aquaculture feed. So now, and Johnathan is really the expert on this, and he can tell you more at the end about things that I've missed in this presentation, Hyojin and I have missed. But this aquaculture really accounts for over half of the world's fish supply that we have in the market. Wild caught fish is really just it's flat now. What's growing is aquaculture, and especially the need for fish oil. Okay, so farm fish needs a protein source, it needs an oil source, and it also needs micronutrients. The traditional way of aquaculture was the farm fish can't make their own meal. They can't make the EPA, DHA type fish oils that we know of as being heart healthy oils. And so traditionally, we harvested low value fish from the ocean, extracted the meal, the oil, and fed that to the high value fish, which is not really a sustainable system. And so now there's really a push for land-based sustainable sources of fish feed. And so soybean meal, for example, is really growing as a source of protein for fish. Okay, and so the work that we do and that Johnathan does is focused on fish oil. Johnathan is the expert in EPA, DHA. And what we're going to talk about in this presentation is the need for astaxanthin. And astaxanthin is a carotenoid that Hyojin will tell you more about. That's important for the color of the flesh of fish. And so not just, this is salmon. And so the consumer expects that the salmon will be a nice pink color, and if the fish were not fed astaxanthin in their diet, the flesh would be white. So fish get the astaxanthin for the flesh coloration from their diet. They eat algae, plankton, and so forth to get the red color. They can't make astaxanthin on their own. And this is actually one of the most expensive components of aquaculture feed. And this is what Hyojin will tell you more about. And before I end, I just want to highlight the work. Hyojin is the leader of this. We've had many people that have worked on the project, and I just want to put a shout out to Johnathan Napier and Rothamsted Research, where we've done field trials between the US and UK and have shared genes across labs. And Ozan Sifsi, who's in our food science and technology department, who has really been instrumental in taking the research from the field to application. And so with that, I will turn it over to Johnathan and Hyojin.
Okay, that's great. Thank you very much, Ed, for that really interesting and comprehensive presentation. I think, it also is important for me to say that the reason that why we at Rothamsted work with Camelina is because you actually suggested it to us as a platform, probably, I guess, nearly 15 years ago. And I think if anybody is listening in that's interested in doing biotechnology in oilseeds, then Camelina really is the plant to try and work with. It's really simple, and as Ed has said in his seminar, he's produced some really nice resources. There's YouTube videos. There's lots of publications on how to transform it. So yeah, it's a really great system. Okay, so let's. So thanks, Ed, also for and as Isabel said right at the start, we'll take questions at the end of the two talks. So we'll take the questions, and they should be submitted through the Q&A function. And then we'll work through those after we hear Hyojin's talk as well. And so I will get my little piece of paper and I will read for Hyojin's introduction. So Hyojin Kim is a senior research associate in the Department of Biochemistry at the University of Nebraska Lincoln, and her work focuses on plant lipid metabolism and metabolic engineering, including developing high oil sorghum for sustainable bioenergy and she has published extensively on plant lipid pathways and supports a collaborative research that advances renewable energy and agricultural innovation. And more importantly than that, Hyojin's been a really great collaborator with us on this astaxanthin project. She's produced some really beautiful results, which I hope she's going to show in her seminar now. So Hyojin, would you like to make your presentation?
Sorry, you're muted.
Sorry, thank you, Isabel. Okay, I think first of all, I want to thank to Johnathan for the kind introduction and thank to Isabel for the kind guidance for the webinar. Okay, my name is Hyojin Kim, and senior research associate in the Edgar Council Lab in University of Nebraska Lincoln. Today, I will present our work on lab to field to application metabolic engineering, focusing on the plant-derived astaxanthin pathway engineered in camelina seed. So to begin, let's look at the global context. Thank you to Ed for the very nice introduction of aquaculture. Yes, fish production and the price of fish oil and meal has been increasing rapidly. And the fish feed contains protein source and essential nutrients such as fish oil and astaxanthin. Before moving on, I want to briefly acknowledge Dr. Johnathan Napier, who is with us today. He is well known for his wonderful work on producing fish oil in camelina, which has a big impact on this field. His work laid much of a foundation for camelina as an oilseed platform. So in summer farming, astaxanthin is indispensable because it serves both nutritional and pigmentation roles. Farm-raised salmon receive the astaxanthin through their feed, giving them the characteristic red or orange color. And compared with other antioxidant, astaxanthin has a significantly higher antioxidant activity and commercial value. Also, the demand and market value of astaxanthin has increased dramatically. So our objective is very clear. Build the camelina as a commercially viable field-ready biofactory that can supply natural astaxanthin to support the rapidly growing aquaculture industry. This goal guides the entire lab to field to application pipeline I will present today. So carotenoids are fat-soluble pigments for the vibrant colors in fruit, vegetables, and seafood. They are also strong antioxidants. Different carotenoids have a different functional group, such as hydroxy group and keto group. When keto group is introduced into a beta ring structure, we recall to this pigment as ketocarotenoids. Astaxanthin is one of ketocarotenoids, has two keto group here and two hydroxy group here. In contrast, other ketocarotenoids typically lack either one of keto or one of hydroxy group. Okay. Thanks to recent advances in biochemistry, metabolic engineering, and metabolomics, we've been able to identify specialized metabolites and figure out how their biosynthetic pathway work. Building on this foundation, the field of synthetic biology has emerged. Synthetic biology now applies the DBTL cycle design, build, test, learn to make metabolic engineering more schematic and efficient. Okay, only a few organisms naturally produce astaxanthin. This includes marine bacteria, algae, yeast, and plant like Adonis. In metabolic engineering, we usually focus on the two main pathways, which are bacteria pathway, CRTW, and CRTG pathway, like this, and plant-derived pathway by CBFD and HBFD pathway. These genes are responsible for adding the hydroxy group and keto groups. And in our work, we aimed to identify which of these pathways would serve as the most effective route for astaxanthin production in plant. To compare the pathways, we used Nicotiana benzamidina as a rapid testing platform. This allows a rapid assessment of pathway efficiency before moving into crop engineering. So TLC and HPLC analysis clearly showed that Adonis pathway produced more astaxanthin with fewer intermediates. But bacterial pathway, CRTW and G pathway, left significant intermediate rather than astaxanthin. This result show that indicates that Adonis pathway shows superior conversion efficiency. So we introduced the P-aster construct into camelina acid. This construct carries the Adonis gene CBFD2 and HBFD1 under a seed-specific promoter so that astaxanthin is produced specifically in the seed. Following the Dr. Johnathan's suggestion, we also added the phytoen synthase gene because phytoen synthase gene is the key control point in the isoprenoid biosynthetic pathway, directing flux toward the phytoen, which is the first committed precursor of carotenoid. By boosting this early flux, we expected to get more efficient astaxanthin production in the seed. As a result, we successfully generated a camelina line that produced astaxanthin, giving the soy oil as a reddish color. So TLC analysis revealed a strong accumulation of ketocarotenoid, particularly astaxanthin, and other intermediates. HPLC analysis also confirmed that astaxanthin as a dominant product exists here. In our previous soybean lines engineering bacterial system, the beta-carotene was left or significantly unconverted. But our astaxanthin camelina line engineering Adonis pathway left minimal beta-carotene remaining, which means efficient conversion from beta-carotene to ketocarotenoid, including astaxanthin and intermediate. So we conducted multi-year, multi-location field trials in both US and UK to evaluate the stability and agronomic performance. These field trials measure the plant growth, seed yield, oil content, ketocarotenoid level, tocopherol level, and germination behavior across different environments. Overall, plant growth showed no visible differences between wild type and Aztec camelina lines. These graphs show the oil content and astaxanthin level. In short, seed yield was also comparable, and only a moderate decrease in oil content was observed. Tocopherol was reduced, likely due to competition for precursors in the isoprenoid biosynthetic pathway. One point worth noting is germination behavior. Astaxanthin camelina lines generally germinated at about 1 day later than the wild type, which caused slightly slower early seedling development. But once they were established, the plants grew normally and reached a similar final biomass and maturity. Next, in collaboration with Dr. Ojan and UNL, we evaluate the functional properties of the astaxanthin-rich camelina oil. They produced the oleogel using sorghum wax, and these oleogels showed a significantly improved oxidative stability during storage period. They also tested the oleogel in the real food application by baking muffins using the astaxanthin-rich oleogel as the fat source. And interestingly, aside from the taste, all performance metrics, texture, structure, stability, they are all excellent. As a result of this work, we got a paper published in Plant Biotechnology Journal, and we are really honored that it was selected for the cover. So from the DBTL cycle 1, we learned that while astaxanthin production was high, intermediate metabolites also accumulated, and the seed performance was somewhat reduced. So moving into the next design phase, we adopt a nature-guided strategy, looking directly at how astaxanthin is naturally synthesized, stored, and stabilized in Adonis petals. This guided the redesign of our next construct and helped us focus on improving both astaxanthin quality and seed performance. So when we looked at Downey's petal under the light microscope, we could see lots of red globular structure inside the cell. These are called globular chromoplasts, and they are where carotenoids like astaxanthin are stored. Next, chemical profiling of Adonis petals revealed that most carotenoids exist in the esterified forms rather than a free type of astaxanthin. In terms of this structure, these astaxanthin esters have fat acid attached to the hydroxy group through the ester bond, which makes them more stable for storage. This natural esterification pattern became an important clue for our next engineering strategy to have a better stability and quality of astaxanthin. Okay, this is general carotenoid biosynthetic pathway found in common plant. And here is astaxanthin biosynthetic pathway found in Adonis plant only, which has not been fully understood so far. Through the transcriptome profiling, we identified 4 candidate genes for biosynthetic enzyme and 2 candidate genes for potential astaxanthin ester synthase enzyme. So we reconstructed the keto-carotenoid pathway modules to understand the enzymatic rules and identify missing components, particularly astaxanthin ester synthase. This became essential component in DBTL cycle 2. So after that, transient assay, we tested the transient assays using co-expression between 4 biosynthetic genes and 2 candidates for Adonis astaxanthin ester synthase gene or tomato autolog gene. This co-expression successfully generated astaxanthin esters form as well as a free type of astaxanthin. In DBTL cycle 2, we built the two type of multi-gene construct. One included only the biosynthetic genes, and the other combined these genes with the astaxanthin ester synthase gene. We used the P2A and T2A peptide linker for coordinated expression while avoiding the repeated use of the same promoter. And for the selection marker, we went with the master gene. Because of the red color of seeds after astaxanthin production, we can simply pick out the transgenic seed without any herbicide application. Camelina engineered with the enhanced construct accumulate a high purity of astaxanthin and produced astaxanthin esters, which improved the quality. The highest astaxanthin content was found in camelina engineered with Adonis ester synthase genes. And importantly, both the seed germination and the seed weight improved significantly compared with the prototype 1. This graph shows the germination result. This orange line represents prototype 1, which shows a delayed germination, but others look normal. This result indicates improvement in both metabolic and agronomic optimization, which is exactly what we aimed for in DBTL cycle 2. So starting in 2025, we began scaling up the processing and testing the plants in the field to check their performance under actual agricultural conditions. And we are really looking forward to working together with our collaborators on the next steps of this research. Then we are now transferring the successful camelina pipeline to soybean, a major global crop. With the collaborator Tom Clemente in UNO, we generated the astaxanthin soybean line, which are currently in field testing. And another example, we are transferring the fish oil biosynthetic pathway into soybean using a modular assembly method such as golden braid or golden gate. Based on what we learned from Dr. Johnathan's work, we designed the EPA modules, and we have now generated soybean lines that produce EPA at around 13% of seed oil. So our long-term vision includes engineering all seed crop to produce fish oil equivalent astaxanthin and other nutritional compounds by using refined and optimized gene stacking strategies. We really want to bring this product to the food, feed, and health market. In conclusion, we found that the plant-derived astaxanthin pathway works better than bacterial pathway. Using camelina as a platform, we are able to move that lab experiment to field applications. The astaxanthin-rich oil we produced showed the real promise for industrial use. And now we are taking the next step by expanding this platform to a major commodity crop, soybean. Altogether, this work demonstrates a complete pipeline from pathway discovery to real-world application. I want to thank our collaborators and partners at the University of Nebraska-Lincoln and Rhode Island Research and funding agencies. That's all from my side. Dr. Edgar Cahoon will take over and go through the following slides.
Yeah, Thank you,
Thank
Hyojin.
you.
I just want to make a couple of points before we end. Maybe, Hyojin, if you could go to the next slide. Okay, so maybe two takeaways from Hyojin's talk. One is that sometimes you have a product idea in mind. In this case, was aquaculture. But what we learn is that actually astaxanthin has many other uses that really have markets, like natural food colorings, uh, also for food applications to extend shelf life by reducing oxidative decay or breakdown of lipids. And so a second thing is that I talked at the beginning about how important knowledge is. And so we started with knowledge that already existed that came from Elizabeth Gantz's lab, Buddy Cunningham, on discovering pathways of astaxanthin synthesis and Adonis. But what we found is that these sort of worked in our first prototype, but didn't work as well as we would like. So that forced us really to gain more knowledge. And so you shouldn't think about application as just being a product. It's also can also contribute to additional knowledge gain. And I want to point out that the work that we're doing with soybean and camelina is really part of a global centers project now that's recently funded by the National Science Foundation and also in Finland. Unfortunately, we couldn't get funding from the UK for Johnathan and his lab to participate, but we're trying to take some of these traits, to food products, to have testing done, and develop new food products through this global centers grant called Food ID, and you can learn more about that at the website. And next slide, Hyojin. This is the last slide, and maybe maybe to stimulate some discussion at the end. So we talked about application and functionality evaluation, but that's not really the end. If you want to have real impact, this is not where you stop. You really stop at trying to generate a product. And so just some things that to keep in mind is that if we want to develop a product, you really have to think about, is there money that can be made? Is the idea that you have really sufficiently valuable that people will buy it, that consumers will accept it? And maybe you should make this decision at the beginning before you start the project if you really are thinking about product development. And especially for transgenesis, not so with breeding, maybe less so with gene editing, but with transgenesis, you really have to think about getting the crop through a regulatory process in your country, in the world. And this is a very costly, time-consuming thing that I wish could be streamlined and be uniform across countries. And also, these output traits that we're talking about are really specialty traits. And in the case of corn and soybean, and wheat, these are large grain crops, large markets that are what we would consider to be commodity. And they have their own way of doing things, they have their own transportation, their own bioprocessing facilities. And you really have to bypass and do identity preservation with these output traits if you really want to get them to market. That's thinking about different ways to transport them that you won't mix with the commodity camelina or soybeans, and bioprocessing that's separate from what's done for the commodity grains. But nevertheless, I think there's real opportunities. And if you're an innovative person, if you want to make money, maybe you can think about this is really what drives innovation, the idea of bioproduct development. And also there's technology that has to be developed along to support this, like in the bioprocessing area. So, don't think about things as just challenges, think about them as opportunities. And if you can do this, you will enhance the bioeconomy of your region, of your country, of the world. And this is capital investment, generating new jobs, having more tax revenue in your locality. And also, we think a lot about how, we want to keep the agricultural sector, we want to keep rural locations, vital. We want to keep, income coming to these rural areas. Too often across the world, we have population that's lost in the rural areas. And this is one way we think of maintaining rural vitality and enhancing value to agriculture. And with that, that's all we have. and we'll stop the share. Hyojin, if you want to stop the share.
Well, can I just say thank you to both of you again. I think that's really, really nice. It's a really nice presentation, Hyojin, of your, as I said, I think your biochemical characterization, your thin layer chromatography TLC is a wonderful thing to behold, and I would imagine that many people watching this are not particularly familiar with TLC because you can't buy it as a kit from Hyojin or Promega. It's it's a skill that you have to hone and learn in the lab, but it's a really powerful biochemical analytical technique. And it's really simple. You don't need to buy a $20,000 HPLC or anything like that. You just need to buy a glass tank and the TLC plates. Anyway, so one thing I just wanted to emphasize, I really liked your last couple of slides, Ed, there that you put up to talk about the importance of translation, and I would sort of completely agree that I think there's a real opportunity for innovators at the moment in a way that wasn't really available when I started my career, as I always say, in the 1860s or whenever it was, a number of years ago. It was much more difficult to take a innovatory, entrepreneurial is the way I look at it, an entrepreneurial approach. And I think, for younger researchers, I think that's an opportunity that wasn't available to me, and I'm quite envious that people have that opportunity now. I would just also point out that I wrote a little sort of opinion piece in Plant Physiology. Okay, don't write in. I know it's not Plant Biotechnology Journal, but it was uh, for a special issue that was put together by Andrew Hansen, who sadly has passed on since that was published. It was a topic that he was really passionate about, which was about translation, but also the issue was about the need for realism in how we address and measure these challenges and problems. So in other words, that the take-home message was, don't try and hype a problem into something so you can just get funded. You need to be realistic about what the opportunity is. And I think Hyojin showed really nicely in her talk about what the opportunities were around astaxanthin. Ed talked really clearly about what the opportunities with biofuels and crops like that. But yeah, the piece I wrote for Plant Physiology was called "How to Eat an Idea," which is sort of like a slightly kooky title, but really was about how do you do exactly the sort of things that we've been talking about in this webinar? How do you go from making a discovery to converting it into an idea, into a product? And I used the example of my omega-3 project, but there are some general rules in that as well. So anyway, enough about me. Yeah, let's. So how are we going to do this Q&A, Isabel? There are some questions in the Q&A. What's the best way to do this?
Yes, I can pick them out from the list.
Okay, and then if you'd like, I can suggest whether Ed or Hyojin should answer them.
Yeah. So the first, I think is very useful question from Pawan Kumar. Dear Edgar, could you please share the transformation protocol for camelina? I think you mentioned that they are already published, and also that you have a number of videos explaining how it works. So
yeah, Tara Nazarenas in my lab, our lab manager, she did a great video on YouTube. Her daughter recorded her, I think, on her iPhone, showing how to do camelina transformation. And I would suggest watching that. I think it's on the slide. If you can look at the slide and see the YouTube.
Yeah,
link.
I can go through the slides because I have the both presentations, and then look for the link, and I will post it here. Okay.
Okay.
I mean, it's a really, really simple. I mean, if I can do it, it's got to be simple, right? And you don't. Do you use the vacuum infiltration, Ed? I don't know whether you still.
We do, but I know that some labs don't.
I mean, we don't. I mean, we don't bother doing vacuum infiltration. So you can be even more lazy like us and just literally dip dip the plants into the agrobacterium solution, and that's it. I mean, it's just, it's almost something akin to a miracle in my book.
Yeah, we find that the most important thing is to have really healthy plants that you use for the transformation. If you have spindly plants, probably won't work very well.
Yeah, and so I think that's true. And I think if you're growing if you like your uh, they're not donor plants, but that you can think of them as the plants that you're going to transform, that if you're growing them in the greenhouse, unless you've got a really super greenhouse with lots of supplementary light, the plants when you grow them in the winter are not going to be quite as fantastic as the ones as you grow in the summer. So you might want to think about growing your plants in CE cabinets over the winter if you're going to transform them.
In our part of the world, it's exactly the opposite. We have the best plants in the winter and the worst plants in the summer. We actually have to use air-conditioned greenhouses in the summer. It's just too hot for us. I guess that's the advantage of the UK. I don't know.
Yeah, well, I mean, it's pitch dark here outside at the moment, and when I was in the greenhouse an hour and a half ago, the lights were coming on, so it's, and all of that costs money. I mean, it's not free.
Yeah, that's true.
My experience in here, vacuum is not necessary thing. I think, yeah, vacuum, when we do a vacuum system, the efficiency of getting a transgenic seed is get high, but sometimes it doesn't need. Sometimes, if you don't want to have much of seed, then you can just do floral dipping method without any vacuum system. It could be work. It could work.
Mm-hmm. Okay.
Yeah, I have included the video link both in the Q&A tool and in the chat box.
Okay, what's the next question?
So next question maybe from Meng Jiang Ling. So I'm wondering if there is any research focused on optimizing oil composition in oat or wheat through breeding for human food applications. If so, which fatty acids or metabolic pathways are typically targeted?
I guess I could try. Generally, okay, so I know that we have a wheat breeding program, and they don't really have, they're not really focused right now on oil quality. They would like to be, but with a lot of these traits, I mean, the major focus is on oleic acid, increasing the amount of oleic acid. It makes for a much more stable oil, really good for food processing applications because you don't have so much oxidation of the oil and off flavors and so forth. So usually, the easiest and maybe the first one that everybody does is making high oleic. But I'm sure Johnathan would love to make wheat with EPA and DHA, maybe. Maybe.
I mean, maybe. I mean, crops like wheat, certainly like cereals like wheat, don't really have very much oil, so you're sort of, you're pushing a rock up a hill, basically. You're trying to engineer the accumulation of whether it's high oleic or EPA or whatever in wheat. Probably ultimately the nutritional value of that would be pretty limited. And this is where the point that Ed made and Hyojin made as well is, this is where you need to understand what your target market, your target application might be. So it's probably from a side, just from an academic research point of view, it's probably interesting enough to engineer the accumulation of a novel fatty acid in wheat or oat. But in terms of making a commercial application, making a commercial product, something that's useful, either economically or societally, you also need to do some sort of techno-economic analysis to understand, really, is this going to be useful? And I would argue it's probably from an economic point of view, from a functional point of view, from a nutritional point of view, changing the fatty acid composition in wheat is probably not necessarily going to deliver significant benefits ultimately to the consumer. So that's for wheat. I think in oat, maybe it's a little bit different because oat is a much more oily cereal. And I know actually some colleagues here at Rothamsted were trying, in collaboration with colleagues in Sweden, were trying to modify the fatty acid profile of wheat. So of oat. So in the case of oat, I think there is some benefit of trying to change maybe make that profile more nutritionist. We've never tried to do that with the omega-3 EPA trait because, as Ed knows very well, it's quite complicated. You need to put a load of genes in, and yeah, it's just hard. Okay.
Yeah. So move to the next question. It's also a question about commercialization and the regulation. One of the key challenges that slow down or at least make it very selective in which trades we're moving forward to commercialization is the cost of the regulation. This may explain why most big biotech companies are focusing on agronomic traits. What is your vision moving forward in terms of application of AI in new trade discovery and the regulation? Will it help reduce cost of entry for new trades, or what we should do to get to that benefit? By Dan Lee.
I have the same response as Johnathan.
Yeah.
That's
I
interesting.
got thrown by the role of AI in that. I mean, I think I'll say my little bit first while Ed thinks. I mean, I think you can imagine that using some of these, helpful tools can help you compile the fairly big dossiers that you need for regulatory approval, but you still will need the data to support your dossier. I think also, I was just reading, I think the regulatory pathway in the US seems to have sort of maybe gone slightly backwards recently in the secure rule that is so, for example, my camelina was approved under the secure rule, and that's been rolled back. So it's yeah, it's the perception is that it's the big companies are the only people that are doing this sort of taking plants through the regulatory approval process. I don't think that's true anymore. It hasn't been for a while. And Kathy Martin's purple tomato is another example that's gone through the regulatory approval process. You know, but it will cost you time and it will cost you money to get through regulatory approval, whatever. So again, it all comes back to this techno-economic analysis. If the value of what you're trying to make is marginal, then actually that's going to be the barrier that will prevent you getting moving forward, because if you can't raise money from other investors because they realize you're only sort of going to generate them an extra 10 cents on every $100 or whatever, then it's going to be really tough. But I've given Ed now a chance to formulate his much more coherent and focused response. Over to you.
Thank you. Maybe I should give it to Hyojin, but okay, with AI, you hear a lot about, like with humans, you hear about digital twins. So I don't know if you can make a digital twin of a camelina seed or a soybean seed. If you had enough knowledge about metabolism and all of this, which is we don't have, but maybe it could be more predictive, like what the metabolic outcome will be using AI tools. I don't know. That would be a great thing if we could just in silico do part of our regulatory package with some prediction about what the impact would be on the metabolism, and to help consumers have confidence that we're not making some deadly allergen in our engineered seed.
And that's a key thing, really, isn't it? It's you have to ensure that ultimately the regulatory packages, which may be a bit of a chore, they're there for a reason, and their primary reason there is to ensure that the consumer has a safe product. And I think ultimately, we're still a long way away off any other approach other than actually you just testing these things to ensure that they are safe. I'm not sure how well the consumer would accept something that says, I haven't tested this directly, but my digital twin tells me that it's fine.
It's a thought.
But
We can be hopeful.
Well, I mean, you have to be. I mean, you have to be hopeful. I mean, as you know, I'm an incredible optimist.
I
But
think
it's
we
a good.
become
It's
less optimistic as we get older,
well, I mean,
sadly.
realistic, optimistic. I don't know. I mean, I think
Yeah.
the question about regulatory approval is, to me, is a, it's a key thing that a lot of the times people in academic research, they don't think about.
Mm.
And the point you made in your talk, Ed, was a really good one, which is that ultimately, the vast bulk of academic research, for them, the finishing line is publishing a paper. And we have a slightly different focus, which is trying to go much further beyond that to get ultimately to a product all the way through the approval process and bring it to market. But there are a number of hurdles that you need to navigate through, of which regulatory approval is one of them. Intellectual property is another one that you need to overcome. And the point you made, which I think is critical, is you need to think about these things at the beginning rather than halfway through your project. Because if you've got to try and retro engineer your project to make it acceptable for regulatory approval, or so it doesn't infringe somebody else's IP, that's a really, really miserable experience. So I'm told.
Yeah. Yeah, I have to agree. I'm not in the business of producing products from plant biotechnology, but I'm in the business of science communication, and that's true. Many scientists think that once the paper is published, all the work is done. So
And last time I checked, you can't eat an academic paper.
no, no,
Well, if times are tough.
Yeah.
very, very tough.
I mean, that's why I called my paper "How to Eat an Idea," but I wasn't suggesting that you actually eat a volume of plant physiology, but even though it's probably highly nutritious,
Little ketchup.
enjoy.
So we have another question from Dan Lee, and I think this one is for Hyojin. I recall the attachant in soybean was developed some time ago at PSI. I think another Korean postdoc, Mr. Park, was working on it. Can you explain side by side the benefit of redoing the proof of concept in Camelina?
Okay, it could be a long, long story. Okay, but yeah, we do a astaxanthin project in camelina and soybean both. But camelina is a really rapid platform rather than soybean. So actually, we have so many vectors having so many different strategy for the astaxanthin production. We applied these vectors to the camelina and soybean. But camelina system is really wonderful. It's really helpful to get knowledge really quickly. So we learn again, learn and redesign, learn,
Mm.
redesign. Yeah, after this repeatable frame sheet, we got a really good strategy so far. So now we are applying this strategy to the soybean. So yeah, and Mr. Park is one of our co-author in this biotechnology journal, and he is also give effort for the soybean and camelina work. Yeah, so beginning 2024, we are finally going to the field evaluation step using a soybean seed. Yeah, that is really that could be a really long story, but I talked really short. Maybe Eddie can add
Yeah,
some
I
comment
can add
or
to that.
explanation.
I think Hyojin might have been confused that Dr. Park is also her husband, but the original work, Dr. Park
Oh,
is a different person.
oh, yeah, right.
Yeah.
Sorry.
So in the earlier paper with Dr. Park, first Dr. Park, uh, what we did, we used the bacterial pathway and we
Yes,
made a lot of beta carotene. So these
yes,
were really orange seeds and not red seeds. And so what Hyojin learned is that the bacterial pathway isn't really in our hands, is not very good for oil seeds. It's this Adonis pathway that really works great for getting very little beta carotene and a lot of phytocarotenoids. And if you had fed the seeds from that earlier iteration, you probably would have orange salmon rather than red salmon.
So moving on, we have another question on commercialization by Adam Gannon. What major companies are operating in the space between academia and production of commercial products? It seems like there is a gap in standardization and optimization processes, as Dr. Cahoon mentioned. Are there private institutions that move forward with translating metabolic engineering work to the field after developing successful transformation strategies?
Well, I guess the most famous example now is Kathy Martin with her purple tomatoes. And, that was a nice paper some years ago, and using transcription factors to upregulate the anthocyanin pathway in tomato. And after many years, she worked through the whole process of taking that concept through regulatory and to market. And you can now buy purple tomato seeds. Maybe Johnathan can really add to that.
Yeah, I mean, yeah, I was at event in Westminster in the Parliament in the UK a couple of weeks ago, and actually there was a really nice presentation. They were talking about, the bottlenecks of how you get, again, we've talked about this a lot actually in this webinar, how you translate something, and it is a bottleneck. There's lots and lots of discoveries. There's thousands of papers that are being published every year with, but what could be potentially interesting innovations and discoveries, but actually hardly any of them get translated. And to me, that's the bigger concern, because we're pouring millions, if not billions, of taxpayers' pounds and dollars into funding basic research, discovering things, and the default seems to be not to translate the interesting things or useful things you've discovered, but the default seems to be to try and discover more things. We're sort of locked into this cycle of, this cult of novelty where we go on and on and on trying to find new stuff, rather than saying, well, hang on a second, we've discovered that, I don't know, you can make astaxanthin in transgenic plants, and astaxanthin could be a really useful, is a necessary ingredient for both aquaculture, for fish farming, but also as an ingredient in foods to make foods more nutritious and blah blah blah. So why don't we translate that? But actually, the default seems to be usually to say, okay, well, you found astaxanthin, so can you go away and find something new, something different? And that's the sort of problem I think that we have, as a community, that we're sort of locked into this sort of almost concept that novelty, there is merit in novelty in itself. Well, actually in patents, the key thing for a patent, it has to be novel, but it also has to have utility. So, and I think that's something we as a community would be better off focused on making sure that we combine utility with novelty. But that's, okay, so that's my unfortunately that's my personal prejudice. And but I also think it's true. So I don't know whether you've got and Ed, about 30 minutes ago, Ed asked me about the purple tomato before I started talking about my particular hobby horse. But yeah, I think the purple tomato is a fantastic example, but it's exactly as Ed said. It was a really gifted researcher in the UK, having doing some basic research, trying to understand the transcription factors that regulate anthocyanin biosynthesis, and then she realized that once she get it's like your slide at the start of your talk, Ed. Once she had the knowledge to understand how to manipulate something, then she applied it to make a black tomato, which is rich in these anthocyanins, which could then be validated using animal feeding trials. Now she's done human feeding trials, and they've taken them all the way through the regulatory approval process. And you're right, in the US, you can buy those seeds. So you can buy, I think, a bag of 10 seeds for a purple tomato. I mean, I think they want $20 for them, which seems, I mean, I think maybe we should start selling our astaxanthin seeds, at a bargain, maybe 19.99 or something like that, as a cheaper than the purple tomatoes. But anyway, I'm joking, right? But yeah, that's all I've got to say, whatever I was saying.
Do you want to add something, Edgar?
No, Johnathan covered it very nicely.
Yeah,
We should stop while we're ahead.
exactly.
So no, we have another question by Pawan Kumar, and this one is also for Hyojin. What other parameters of substantial equivalence in the astaxanthin camelina lines did you measure besides oil content and seed germination? Additionally, in how many other parameters did the lines show reductions related to substantial equivalence?
I think I have a little bit answers. Okay, we measured the yield and we found no differences were observed. And aside from oil content and seed germination pattern behavior, as mentioned, that the lines show the slower early post-germination growth, and also they have a reduced seed weight. That's all. That's all we tested. We measured.
What about the tocopherol and also in the soybeans? I think we measured ABA content.
Oh yeah, but in the camelina seed, we don't have any differences of ABA content. But when we measure the ABA level in astaxanthin soybean line, we have seen some reduced amount of ABA during the seed development. But we didn't mention about this one today.
Yeah. For the biochemist, if you know about the plastid isoprenoid pathway, geranyl diphosphate, which is the precursor for making beta carotenes, also the precursor for tocopherols, also beta carotenes converted into ABA. So there's a lot of branch points, and you got to be careful about manipulating the carotenoid pathway so you don't get these off-target
Yes,
traits.
also upper maybe from the GGPP, that is also precursor for the tocopherol tocopherol product. So yeah, that's why we saw the reduced tocopherol level in the camelina seed.
And tocopherol is basically vitamin E, for those who don't know.
I think you have started answering the next question already. It's from Shay Uniwo. Again from Hyojin. Great presentation. Have you considered metabolic trade-offs and how the enhancement in astaxanthin content affects composition of the plant in terms of other nutrients? I think the answer is yes.
Yes, and to enhancement of astaxanthin production, we added the phytoin synthase gene based on Dr. Johnathan's suggestion. So yeah, that is a really good point to get the really good result in our study. Yeah.
But I think our second iteration where for whatever reason, this is still knowledge that I think we need to gain, is that if you can sort of store away astaxanthin as these esters, somehow it has less impact on the plant. So I think it tells us that in general, if you can package something in a plant cell and put it away in a place maybe that's less toxic to the cell, that's always a good thing.
Yeah, they must be isolated.
So next question. Have you done any work on plant species like Moringa oleifera? Any of you?
No, but I would say that we're agnostic. I mean, if there's funding to be had to work on anything, we're willing to do it. But we've also worked on cassava in the past and made orange cassava, and that was published in a Plant Biotechnology Journal a few years back.
What a great journal.
Yes, it's a fantastic journal, and most importantly, a great editor-in-chief.
I've heard that said.
So there is a practical question for Daniel Hagos. For Camelina, what do you use to protect from insect pests, including ants? In the field trials, I assume, or maybe in the greenhouse.
I have to say that, and we haven't really had insect problems in Nebraska. I think either in the greenhouse, I mean, we've had maybe some fungal disease in the greenhouse, but surprisingly, it's been very pest resistant, both microbial and insect in our field trials.
Yeah, I mean, I think in our field trials we have had some. Actually, this year was the worst year that we have with cabbage stem flea beetle, which is but that's a problem in Europe in general because the insecticides that were used to control that the neonicotinoids, whatever they're called, insecticides have been banned in Europe for the last few years. So basically, the insects are getting a free pass, and they normally colonize oilseed rape canola. But if people start growing camelina, which is obviously a brassicaceae, they are quite happy to eat our camelina. We also get problem with pollen beetle as well, which is another brassica type pest. But it's not as bad as the flea beetle.
We've had more problem actually with our soybeans in the field. We have Japanese beetle problems some years.
I mean, this is why, getting into the field is the real gauntlet. It's the real test to find out whether or not your crop is going to survive or not. Because as you said, Ed, growing something in a white box at the same temperature for 365 days a year, which is what a CE cabinet is, bears no resemblance to what it's like in the real world out in the field, and that's the environment that the farmer has got to try and make a living with. So you need a robust crop, and pest pest control, all of those things, agronomy, all of those things are really important components of understanding how to make a biotech crop work in the field.
Mhm.
Also, here we have another practical question about the propagation rate and which X plants are the best for producing a particular metabolite. I assume we stay with the metabolite that you are studying. What are
Could you repeat that question? I didn't quite understand.
Yeah, from Mina Pendey is what is the propagation rate of camelina and which X plants are the best for producing a particular metabolite?
I mean, I guess that, I would say, I mean, just jumping in here while Ed prepares his comprehensive response. I think normally we can get 5 or 6 grams of seed from a single camelina plant. So, you put one seed in the ground and you get, I don't know how many, 1,000 seeds or maybe more. 500 seeds from that one seed. So the multiplication rate, I mean, when we've done sort of Fermi calculations, back of the envelope calculations, if you want to be conservative, you can say that for every one seed that you plant, you will get 200 back in the next generation. So that if you're trying to multiply something up, that you can use that as a sort of simple rule of thumb. Ed?
Sounds good. Sounds good.
Yeah.
And regarding the eggplant, I assume it's the seed.
Yeah, it's the seed.
Yeah.
Yeah.
So we have a question from Farouk Kasim. Thank you for the presentation. How can plant lipid engineering and biofortification research be used to improve agriculture, nutrition, and economic growth in underdeveloped countries specifically?
I think it already is. And, there's funding for like from the Gates Foundation, Harvest Plus, for example, golden rice. I have to say, at the end of the day, I think a lot of it comes down to the wealth of an area. I know, this, if you can promote economic development, then people have a more diverse diet. And that's largely what, is the problem with malnutrition. I mean, that's easy for me to say in the US, but yeah, maybe Johnathan wants to say something.
Well, I thought, what are you going to say? I mean, like in the US, the Center for Malnutrition, is that what you're, I mean?
Yeah, well, we actually do have problems with nutrition in the US even.
Yeah. I mean, in all seriousness, I think obviously economics, the economics of what people can afford to spend on food, still sadly dominates, many, several billion people's existence. They're constrained by how much money they've got, which dictates the food they can eat. And so, uh, it in those circumstances, actually the big issue is really just ensuring people have enough calories, per to eat per day. But then once you move beyond that, then you have to think about, well, how can we make the food more nutritious to ensure that it's, appropriate, to ensure that everybody has the correct nutrition that they need at all stages in their life cycle. But these are rightly signposted as global challenges. They're part of the UN Sustainable Development Goals, which we are not going to meet anytime soon.
Right. And
He
for
says,
example, if you have the option of orange yam versus a white yam, I mean, I think there's some education that goes along with that, that you should probably eat the orange yam, especially in areas in developing parts of the world.
yep.
Next, we have a very niche, I would say, a question for Edgar. What do you think of the sphingolipid function in plant pathogen sensing or treatment?
Well, you can read my review article and the plant physiology on that topic, but I think that is an area that that is growing, and I think that's another area that we work on is sphingolipid metabolism. It's well beyond this webinar, but yeah, I think that sphingolipids really play a very important role in controlling programmed cell death and its relation to uh, like the hypersensitive response in plants. Yeah, study that. It's a good area.
Then we have a question from Anirban Roy, and I think I must rephrase it. I think they are asking when you are in at a isolated location geographically, and there is limited market opportunities, how to make the choice about what crop to research and what trait to focus on.
I think you should focus on, the crop that's relevant to your area and do whatever you can with the resources that you have. If you have genetic diversity and you can make crosses, I mean, really capture the genetic diversity through breeding. Yeah, I know that. Yeah, I understand that. It costs a lot of money to do this kind of research, and it may not be available everywhere in the world. But think about your area and how you're going to have impact in your area with the crops that are local to your area, and use what resources are available. Start with breeding if that is, an option.
Mm-hmm. I think that sounds reasonable.
Mm-hmm.
Anything to add, Johnathan, Hyojin?
No, I think that's good, really good advice.
And then we have a couple of questions by Babak Nakoda for both Edgar and Hyojin. How much oil does sorghum have?
Well, in its seed, it's like what Johnathan was talking about with oats and wheat. Not a whole lot, maybe
Mm.
maybe 3% of the total seed weight. But what we're trying to do is engineer the leaves and stems to make oil, which they don't normally do. And so maybe in our best lines, we have maybe 7 to 10% dry weight of oil in the leaves. The stems maybe 3 to 5%. Obviously, we would like to do much more, and that's what I was talking about, how we're using systems biology to really understand the plants that we have now, to understand what bottlenecks, metabolic bottlenecks, and other bottlenecks are really limiting the production of oil. But I would say that making vegetative oils is a different topic, but that's really one of the grand challenges of metabolic engineering, and one that could have a lot of impact. I mean, we're thinking about it for biofuels, but for just having oil for food use, I think it's if you think about how much grass and how much biomass there is in the world, we think about how if you can make 10% of that weight as oil, there would be so much oil in the world that
Yeah.
you could run your car, your jet plane, while still having enough to eat.
What a wonderful world that would be.
Got to
I
stay
know a song
positive.
like that.
So, and the next question, have you tried CRISPR technology for astaxanthin production from algae or other crops?
We haven't, because especially in the case of astaxanthin, this is really a novel pathway, and so like camelina or soybean doesn't have the capacity, doesn't have the genes that are necessary to make astaxanthin. So we've relied on transgenesis. I mean, we have thought about ways if we could understand the enzymology, these enzymes that make astaxanthin, they've evolved from enzymes that are found in all plants. And if we could figure out, structurally what gives rise to different functionalities of the enzymes, maybe someday we could do gene editing and make those modifications very precisely. But that's a day that's long in the future, I believe. But, certainly you can do gene editing maybe to enhance the flux of a metabolite into the pathway, or make a better chassis for metabolic engineering, make a more robust, more oil-rich camelina seed, that type of thing.
Yeah, I mean, I think that's the key point. I mean, most of the traits that we've been talking about this afternoon, they are, require you to add in extra genes by GM, by genetic modification, and they can't be generated by the mutagenesis function of gene editing. So, I mean, and as I always say, although gene editing is sort of presented as the new silver bullet that has superseded boring old GM, actually you can do much more with GM than you can do with gene editing. That's just the hard truth.
Interesting.
Not trying to be controversial here either, by the way. That's just
Maybe we should stop now.
yeah, I was going to say mic drop, boom.
Then we have another question, this time from Lee McLaren, and I think it's a curious one. Is if the Adonis astaxanthin production is more stable than the bacterial pathway, why not introduce the Adonis into bacterial use as a production system?
Actually, we're trying to do that, not with bacteria, but actually in other in yeast. Yeah, yeah, and that might be a way to get to market quicker, because sadly, the regulatory environment for genetically engineered yeast is not at the same level it is for plants.
Mm.
So it means it's easier, a little bit easier,
Easier, but
or faster.
the cost probably would be higher because you can't, scale up the same way in a fermenter as you can in a field. So you're probably looking at more niche markets, higher value markets.
I think, and that's a real key thing that people forget, especially when they're talking about the potential of, single cell solutions, delivered in for engineering biology or whatever using fermentation, is that, if if you want to scale up beyond your 1,000, 5,000, 10,000 liter fermenter, or half, 500,000 liter, whatever it is, those, in terms of actually delivering things that are useful at the scale they're needed, like to feed 8 billion people, you're basically having to reinvent a whole new level of technology that doesn't exist. Or the alternative is you can use agriculture, which is what we've been using to feed the world for 10,000 years. And the infrastructure exists for doing that, for growing at scale, for processing at scale, for moving stuff around the planet. That all exists. So why not use agriculture rather than, do something different? But that comes back to this cult of novelty where you have to be seen to be doing something different. And that, I think, is really self-defeating.
And people don't always realize that, the bacteria and yeast in a fermenter, they have to eat also.
Exactly.
And so you typically would feed them glucose, dextrose, isolate it from cornstarch. Yeah.
Yeah, that's right. So, you're relying on agriculture one way or the other. And the fermenter, it's for its carbon source. It was relying on cane sugar or whatever it was to provide the energy for the cells to grow. So, and it drives me nuts because those are the sort of things that are missed out of the life cycle analysis that lots of people do to demonstrate the economic viability of products that are produced by single cell fermentation or whatever. But nothing's for free. There is no such thing as a free lunch.
Photosynthesis? There's cost to that also.
Yeah, agriculture is not free either.
Yeah, it's right. It's true.
But it's cheaper. Support our great British farmers and
I really enjoyed this question and the discussion about the cycle and how we always go back to agriculture in the end and the plants. So next, there is another question from Dan Lee. Another follow-up question. I know you may not have time to reach to this one, so we did have time. Is there a way or a trade we do not need economic of scale to make it profitable? I think I know the answer, but I will leave it up to you.
Is there a trait that you don't need?
Economic,
Say that.
Economy of scale.
Oh, well, there's like a pyramid of value, and usually at the top it's like pharmaceuticals and like vaccines and drugs, that type of thing that are really valuable that you don't need very much of. And there are people who are doing great work in like plant cell cultures to develop vaccines and drugs. Yeah.
It's not
That's
really
at the top of the pyramid of value.
that it's more a metabolite. Yes, that could be a trait. So how are you all feeling?
Great.
Great. And then we have 5 questions and we have, I think we have time, 15 minutes. So I don't know.
Okay, let's do it.
Answer short.
Yeah, I won't say anything.
So we have a question from Rafia Norulain. Sorry for butchering all your names. How can genetic engineering help combat recent climate disasters in case of floodplain agriculture, and to what extent? What do you suggest is the best way to approach this problem?
Well, I think it already is, and a lot of it's from breeding, from looking for genetic diversity within crops that allow, some varieties to do better with less water or better with heat, and trying to introgress those traits into crops through breeding. But certainly, there are many traits. I mean, insect, microbial, pest tolerance that I think can be very easily addressed with either maybe gene editing or transgenesis. Johnathan, why don't you take over?
Yeah, it's complicated, I believe, is the standard answer for something like that. I mean, yeah, I mean, yeah, there's no one easy answer to there's no one easy solution to all of this. It's sort of, I feel that it's like one of these, it's a global challenge that needs a sort of concerted and collective effort, analogous, I use the example of the COVID-19 pandemic. The reason why we're back to some sort of normal normal societal behavior and not wearing masks and locked in our apartments is because actually science was able to come up with a solution in pretty quick order with the RNA vaccines and develop them and test them and scale them up. So to me, that sort of is the beacon of hope for trying to fix these bigger challenges, because I think climate change, developing crops that are climate resilient, is a much bigger challenge than generating a vaccine to COVID-19 or something. It's an order of magnitude, maybe more complex, and it's a sort of shifting, ever worsening issue. So, I think it requires some sort of concerted effort, which we don't have at the moment. So that's not an answer. It's just pointing out we've got a big problem. Sorry.
Not sorry. Don't be sorry.
Yeah, well.
So next we have a long question by a PhD student named Hamid Gul. They are finding some problems when doing their research. So I'm going to we are using CRISPR-Cas to study the root architecture gene. In our first trial, field trial with a segregating T1 population, we observe a significant and unexpected drag on yield. And they are working on maize. In your experience, what is the most critical first step to diagnose this? Should we prioritize screening for a background effects like somaclonal variation, or focus on advancing generations to isolate the homozygous line and see if the yield penalty persists?
I think for us, we would do like a systems biology approach, and we would do all of the omics and see what's different between the edited and non-edited lines as a start. Omics are so cheap, especially transcriptomics and all. It's an easy thing to test, but it may not lead you, to the right answer. But these other things are just you're just testing things kind of randomly. At least with systems biology, you get some real data that you can use as a basis for making more informed decisions about how you address this problem. But it is a problem, that Johnathan has highlighted, that taking things to field, you learn things that you thought your idea was great, in the lab, but when you go to the field, there's something else going on, and it could be any reason. I mean, you might want to even take that line and go to a different location. Maybe there's something in your environment, the soil or something, that is causing the problem. I don't know. It's
I think yield drag is the norm. I mean, I think this is the problem, is that people imagine that that you make a transgenic intervention especially in metabolism, and your host chassis organism, whatever, is just going to shrug it off and will not be perturbed by this. But you are perturbing the system, and so actually, I think seeing a penalty of some form or other, metabolic, phenotypic, whatever you want, is entirely predictable. I mean, it's entirely expectable. It's not you can't predict what's going to happen usually, but it's perfectly normal to see it happening, I think. I mean, so that's why another reason why probably the simple thing to do also is screen a large number of events, because of the plasticity in plants and their metabolism and their genomes, they're actually well adapted to trying to mitigate the perturbations that you're engineering. So screen lots of events is the simple way to start with, and then you can do some omics as well when you've done that.
Yeah. I mean, you may have off targets, who knows? And that's why looking at a number of events is a good thing.
Yeah.
And a breeder would tell you, just cross it into the elite germplasm, and that will solve everything.
That's right. They would say that, wouldn't they?
They would.
Yes.
And they might be right.
They might be right.
Then next we have a question by Venkata Partha. Since the Adonis CBFD2 and HBFD1 cassette functions well in transiently infiltrated leaves of Nicotiana ventamiana, would it be also feasible to express this pathway in leafy vegetables to enhance their nutritional value, or is its effective use demonstrated only in seed-specific applications, such as the camelina system?
Actually, there's been a lot of work done on astaxanthin synthetic biology and engineering, and it's not so much with the Adonis pathway, but there's already some good literature on making astaxanthin in crops like tomato fruits. So it's kind of already been done to some extent, maybe not with the Adonis pathway, and that should be probably tried in some engineering study. But again, we get back to whether is there a market for red lettuce? So, I mean, as a consumer, are you willing to pay maybe twice as much to have a red lettuce versus a green lettuce? I don't think you will be. Yeah, who knows? You have to do your market research and see whether that really is a viable product.
And recently, a paper published this year reported astaxanthin production from the Adonis pathway in tobacco leaves, which demonstrate that the expression of Adonis pathway in the leaf tissues is visible. Yeah, indeed, it's visible.
Our experience with beta carotene was that if we could produce really large amounts, like in cassava leaves, but then that affected photosynthesis a lot. And so sometimes with these pigments, you might have interference with photosynthesis, and that's something also to consider.
So only 2 questions to go. From Sami Madahana. Thank you for the presentation. Have you tried using gene cassettes in controlling wheat stem rusts?
We have not.
UG99 variants.
We have not.
You have not?
We're not really a plant pathogen lab. It sounds like a great idea, and I would encourage you to pursue it.
Last question. Here, at least in the Q&A tool, maybe there are more in the chat. Can we envisage using bioengineered plants as a sustainable source of natural dyes, as an alternative to synthetic dyes? And what do you think would be the main obstacles to overcoming?
I think that's a fantastic question, and a good question to end on, maybe. That there is, throughout the world, I think there's a big push to get rid of chemical dyes in food products. And so I think there's a huge emphasis now on finding natural pigments. And in the US, I think Heinz, that makes ketchup and these other products, I think they have released some statement that by a certain year that they're going to get rid of all the chemical dyes from their food products. And so there is definitely a market for natural food coloring. I think the challenge is to find the right colors. There's only so many colors in nature, and you can only, maybe you can't exactly get the green color or the blue color that you want in your food product with natural dyes. So you might have to be willing to accept some other color.
And I think, the key thing is going to be scale. So if you think about, the amount of, I don't know, if we think about colors and dyes in foodstuffs, like, in your tomato ketchup, the volume that you might need is going to be pretty enormous. So again, you'll need to think what's the most economic way to produce that. Let's think about that. Oh yeah, agriculture. So that's the way we do it, I think. And this time next year, we'll all be astaxanthin millionaires.
Billionaires.
Yeah, sorry, my mistake.
A million doesn't go very far anymore.
It really doesn't, no.
Inflation.
So that was the last question. And
Yes.
yeah, we are 3 minutes short from being here 2 hours. So thank you everyone who has stayed for this long of the attendees, and of course our speakers and chair. Thank you all. And I will do a little bit of our publicity of our upcoming activities. We have a next webinar next month in December on cassava breeding. So I will post here in the chat the information just in case you are interested in attending. We will share with you all the recording of this webinar on YouTube. And thank you for staying this long with us and for answering all these questions. Thank you,
Thank
Hyojin,
you.
Edgar, and Johnathan.
Thank you very
Thank you
much.
so
Yeah,
much.
thank you. It's been fun.
Yeah,
Thank
on
you.
behalf of Plant Biotechnology Journal, thanks to Ed and Hyojin for their talks, and thanks to Isabel for hosting.
Okay.
Perfect. We can repeat sometime.
Okay.
For sure.
Okay. Bye-bye.
Okay.
Bye, everyone.
Bye-bye.
Bye-bye.